So this is the structure. This is the full thyroid follicle, and we've enlarged this portion. So this is the enlarged portion, and you can see that this is the sodium iodide transporter. Iodine comes through the blood to the thyroglobulin, and it's co-transported together with sodium. So it's a symport. So the transport is towards the same direction of two molecules. The iodine then diffuses through the cell and enters into the colloidal region with the help of the Pendrin channels, where the iodine is converted to oxidized iodine, right? And the oxidized iodine undergoes iodination of, that is, the tyrosine undergoes iodination with oxidized iodine. And in the nucleus, with the help of the endoplasmic reticulum, rough endoplasmic reticulum, the thyroglobulin is synthesized and it's secreted with the help of secretory vesicles, exocytosed out of the cell into the follicle or the colloids of the follicle, and inside the colloid it undergoes iodination, where the iodine molecules are added to the tyrosine residues on the thyroglobulin. And then the second step is conjugation, where two iodine molecules conjugate with each other. And finally, there is endocytosis, where the uh, thyroglobulin with conjugated iodinated tyrosine residues are endocytosed into the follicular cell, and it undergoes proteolysis so that you get triiodothyronine and tetraiodothyronine or thyroxine, and it is released into the blood through the channels. In this figure, you can see the follicular cells. This is the colloidal region, and this is the capillary region. And the blood vessels are present in here. So, in the first step, the rough endoplasmic reticulum synthesizes and packages or transports and packages thyroglobulin into secretory vesicles, which can fuse with the colloidal membrane or the apical membrane of the follicular cell, and release the thyroglobulin into the colloidal region. The second step, you can see that the blood carries iodide towards the follicular cells, and the iodine is transported into the cell through the sodium iodide transporter or the NIS system, and the iodide then travels towards the apical membrane, and it is released into the colloidal system with the help of iodide fluoride transporter called Pendrin, and the thyroid peroxidase converts iodide to oxidized iodine. In the fourth step, the thyroglobulin or the tyrosine residues in the thyroglobulin undergoes iodination and forms pre-T3 and pre-T4. In the fifth step, the pre-T3 and the pre-T4 is endocytosed into the cell, and it fuses with the lysosomes so that it undergoes proteolysis. The protein undergoes proteolysis, and you get active T3 T4 residues inside the secretory granules, which are then exocytosed into the blood circulation. So these are the different forms that are available. You have monoiodothyronine, diiodothyronine, and a monoiodothyronine and a diiodothyronine combines together to form the pre-T3, whereas two diiodothyronins combine together to form the pre-T4. And this is the structure of T3 and T4. So the sodium iodide importer or the NIS transporter, it's a co-transporter. It's a glycoprotein with 13 transmembrane domains present on the basolateral membrane, and it transports 2 sodium for each iodide into the follicular cells. Active transport of iodine against the concentration gradient is driven by the sodium transport down its concentration gradient, because we know that sodium concentration is higher in the extracellular fluid and lower in the intracellular fluid. So when sodium travels down its concentration gradient from its higher concentration in the extracellular fluid towards the interior of the cell where it has a lower concentration, this energy is coupled to the transport of iodide molecules from the outside of the cell where it is in lower concentration to the inside of the cell where it is in higher concentration. Once inside, the iodide diffuses towards the apical membrane, and the iodide is converted to iodinium, that is iodine plus, by the peroxidase enzyme, and the iodinium iodinates tyrosine residues of the thyroglobulin, which is present in the colloid. So this is the structure of the sodium iodide transporter. As you see, there is 13 transmembrane domains, and there are extracellular domains also. And in this structure, sorry, in this figure, and in this figure, you can see that for every 2 sodium molecules, 1 iodide molecule is transported into the cell, and the sodium concentration is maintained by transporting the sodium out of the cell and the potassium into the cell by the sodium potassium ATPase. So this sodium potassium ATPase ensures that the sodium concentration inside the cell does not increase. So Pendrin is an anion exchange protein found in the thyroid, ear and the kidney, and it was initially identified as a sodium independent chloride iodide exchanger. Also, it can also accept formate and bicarbonate as substrates. So in the thyroid, pendrin mediates the efflux of iodine across the apical membrane of the follicular cell. So in this figure, you can see thyroid, ear and kidney. So in different regions, you have the NIS system in the thyroid, where sodium iodide is co-transported into the thyroid follicle cells, and pendrin will release the iodine from inside the cell to the colloidal region. In the ear, you have another enzyme called the AE2, another transport system called the AE2, where chloride is antiported for bicarbonate ion, and the chloride and bicarbonate is again antiported by the pendrin into the endo-lymph liquid. Third is in the kidney. Uh, Third is in the kidney. You have H plus ATPase system, which transports proton ions, and the pendrin transports bicarbonate for chloride in an antiport mechanism, which forms the urine. Okay, so these are the different like locations or different functions of pendrin that is present. As far as thyroid hormones are concerned, we are concerned with this particular function. So the thyroid peroxidase is also called TPO, and it's expressed in thyroid and secreted into the colloidal solution, and it oxidizes iodide ions to form iodine atoms for addition onto the tyrosine residues on thyroglobulin. Human TPO is encoded by the TPO gene. So in this structure, You have the tyrosine molecule with plus an iodide plus a proton plus water. It gets converted to the monoiodinated tyrosine plus water. Here again you can see diiodotyrosine